Hi. Unfortunately, I have symptoms of a cold, and I'm going to a professional in about an hour, and there's some chance they won't let me be among the living. So I didn't want to do a recording on this chapter, although it's an easy, good chapter to do. Um, Fear and stress are part of the topics that I've done my research on. The opening vignette is a good one. Um, must be really interesting to have that level of loss of fear. There was just another really famous case of this happening that was revealed over the last year. It was in the news. Um, Within limits, of course, fear is good for us. The early part of the chapter overviews lots of different types of emotions and reintroduces us to the sympathetic and parasympathetic nervous system, theories of emotions. James Lang was definitely a popular one, and Walter Buchanan rears his head in here again. So, be able to recognize these three on page 469: folk psychology, common wisdom, what's going on, James Lang, what's going on, Canon Bard, what's going on. And shortly, the book discusses cognitive attribution and Schachter, um, which is more modern thinking on the topic. And here we have people getting injected with epinephrine and then given a model that's either happy or angry, and the same physiological stimulus, the epinephrine, paired with a happy confederate, you are happier, and angry, you're angrier. So there's the Schachter cognitive attribution model, which is arguably the best supported by current data. So know that one. Polygraphs, lie detectors. The book uh, discusses this, looks at the data. My brother used to be subjected to these, and occasionally the operator would just decide that something wasn't right, and my brother was always in a state of terror over these things. Aldrich Ames is discussed here, longtime CIA agent. I believe he has local ties, uh, and he used to be able to defeat this. So I'm not a big fan. Neither is your author, Mark Breedlove, a big fan of lie detectors. Um, there's Darwin. He was a fan of emotions, and he wanted to know, about the evolution of emotional expression. And there's some fairly intense figures there from his publication. Uh, Crested black macaque, placid, although still pretty intense. I wouldn't mess with that animal. Um, yeah, the same when pleased by being caressed. That doesn't look happy either. I guess I don't like black macaques. So evolutionary psychology. I used to be a huge fan of that. Less so these days. I like the European approach to this topic, but certainly it's relevant. Facial expressions in different species, uh, response stereotypes, and soon we're going to get to somebody I used to like and then didn't. Oh, this is really interesting. Um, yeah, this is predictive of long-term issues in people, shy and having phobias. This is one of the topics that Dr. Karpiak was trying to do research in when we had mice. I was looking at the effects of exercise. She was looking at the effects of giving them toys and small challenges, because there was research in people that children who overcame small challenges became less shy and more able to deal with people. Uh, that's really pretty profound. And certainly don't try to memorize this, but take a look at it and appreciate that there's a spectrum and there's intensities, and but don't memorize it. Ekman. I used to be a huge fan of Ekman. Less so now. I think he's gone for the Bucks. And he used to publish a lot of stuff and did good science. And now he's selling uh, kits to the Homeland Security and CIA and police stations to try to figure out whether people are showing micro expressions and determining whether they're telling the truth or not. More faces, and the black line here, level of agreement by chance. And you can see that things look pretty universal if you're looking at Western literate and non-Western literate, but isolate non-literate, there are exceptions, particularly surprise and disgust. Disgust, recognition of that is not really much different than chance. And this is one of the things that Darwin was interested in, and it's still being researched. Um, I suspect that this book was published, this edition was published around the time of the last election, and I think Mark expected Hillary to win because he usually puts a picture of the president in here. And so, yeah, there's an interesting look at a picture where people with different perspectives on politics and people would interpret that face differently. Um, Here's a pleasant picture of deep and superficial branches of the facial nerve affecting facial expressions. And I believe on the next page, I had a friend who had this. Oh, no. Okay. Let me do this before we go on to the friend. This research really swept the nation, swept the world, that holding pencils in laboratory research, if you held the pencil on the way you do in the left picture, that's not mimicking any emotions. But if you hold the pencil the way you're holding it in the right picture, you have a happy face. And the story was that your systematic objective assessments of happiness are higher on the right figure. And that's not holding up too well in the literature. I have the new book. I'll have to look and see whether this survives. Um, there's Bell's palsy. Um, it can, when it's fresh, it's pretty dramatic. Typically there's a great deal of recovery, sometimes complete recovery. But I had friends; they were identical twins, but you could tell them apart because one twin had Bell's palsy in her childhood, and one had not. And one had like a quirky smile, and she was the one who had Bell's palsy. Always shows up in here. Okay, there's a figure to die for. Look at that and tell me what kind of a brain section is that? How is the brain cut? It's a sagittal section. More specifically, it's a mid-sagittal section. You can see the pretty humongous massa intermedia here with the third ventricle going around it, and then the cerebral aqueduct, and back to the fourth ventricle. And boy, there's a lot of nuclear groups in there that we spent some time talking about. So lollipops, the solid balls are where the cells are that produce the neurotransmitters. And then the terminals don't have little claw feet, but they have arrows. So let's start here, which we've seen in the sleep chapter, locus coeruleus, painted blue appropriately, passing rostrally through a part of the brain I know and love. And then you have the substantia nigra. Perhaps you'd prefer that be black, but that's in the midbrain. And dopamine, and then the VTA, ventral tegmental area, also dopamine. Notice that those axons run together and they join up with locus coeruleus axons and then go to the forebrain. Where do they go? Many places, but one thing you should always think of when you think of the VTA, you should also think of the nucleus accumbens. And there it is. And this place where they all get together is the medial, suggesting it's not lateral, forebrain, because that is the forebrain bundle. It's a bundle of axons. And self-stimulation is down here, brain self-stimulation. If you wanted to stick an electrode into a rat and have a high probability that the rat would press a lever to get electrical stimulation from that electrode, you would go for the MFB, the medial forebrain bundle. It's complex. Electricity is stupid. Axons are stupid. You stimulate here. You're going to generate axons that release neurotransmitter into the forebrain, but you'll also generate axons that burn back, and, for example, could end up sending noradrenergic input into the cerebellum. So it's really interesting, and I wish I had a video of this for you, but a lab that I worked next to at UCLA used to give rats cocaine, and boy, there's this certain look they have when they're all coked up. Their heads are nodding and they're bobbing, and when they're pressing a lever for self-stimulation, it looks very much the same. It's a very intense stimulation. So know the media forebrain bundle, know those areas that we've already done. Olds and Milner, they're the ones who did this way back in 1954. They found it by accident. They were doing arousal research. And they knew that electrodes in some parts of the brain were aversive, and they didn't want animals getting stimulation that was aversive. So they put the animals in an open field, which is a field that's open. It has walls, of course. And they divided it up, possibly into quadrants, certainly into halves. And when the animal was in one part of the open field, they stimulated the brain. And they figured, well, if they don't like it, they won't come back. And if they don't come back, then they're eliminated from the study. But they found a rat that kept coming back for more. And allegedly they lost the electrode. They didn't know where it ended up. But they had been trying to stimulate sort of the reticular formation down here. But I think the electrode sort of got dinged as it went through the hole in the skull and was deflected rostrally. But the rest is history. Decortate rage. My first surgery. Trying to create a behavioral state that's very high in aggression, non-targeted aggression. And boy, were we scared when that rat was waking up. But we missed, and he did not exhibit any rage. You look at PAPEZ, and of course you can conclude that that's Pepez, which I told you for 20 years it was, but Mark Breedlove knows more than I. It's capes. Rhymes with capes. So it's papes. P-A-P-E-Z, papes. The papes circuit. And yeah, there's a lot of the parts. Later, Paul MacLean, 1949, the year I was born, so it's still a long time ago. Here's a figure in your textbook. I won't test you on it, but the elements of the Papes circuit are here. Notice, by the way, that there's this little island here that's been cut. What kind of section is this? How has the brain been cut? Sagittal, mid-sagittal, just like the self-stimulation rat brain that we saw a page or two ago. But in the rat brain, that circle was way bigger. And the blue liquid that isn't drawn blue here had to go around it. It's a big island. Here, it's just a little nubby. That's the massa intermedia. And I keep saying, some of you don't have that. Your left and right thalami don't touch. And of course, if this is your thalamus, then below it is the hypothalamus. What's of interest here? Well, buried in the temporal lobe, you have the amygdala, although I prefer to say amygdaloid complex, and you have the beginning of the hippocampus, but notice it's kind of long and it disappears as it moves out of sight here. And when you think about the hippocampus, you're supposed to think about seahorses, hippocampus. I like to think about bananas and the letter C. So the forebrain, as I said long ago, is brought to you by the letter C, and the hippocampus sort of looks like a banana, and it's curving up and around. And speaking of curving, this structure here, which is appropriately labeled as the fornix, see it? There's the fornix. The fibers originate in the hippocampus. They come out into the fornix. They do this weird. They go up, they go forward, they go down, they go back, and they terminate in the mammillary body. Now, the mammillary body is part of the hypothalamus. It's below the thalamus. How far below is it? It's the outside of the brain. So if you're in the lab, you've seen the mammillary body. Although in sheep it tends to be one bump. In us, it's clearly two bumps. And then you have a pathway out of the mammillary body to the anterior nucleus of the dorsal thalamus, or to friends, anterior thalamus. And then from there, you have fibers that go up to the cingulate gyrus, and the cingulate gyrus keeps coming up, and it'll come up for the rest of the course. And then fibers arc back and around, and eventually go to the hippocampus. That's the Papes circuit. Now, I have, thinking I might be sick, I have crafted an image for this. So if you go to, where'd it go? Why do I always have trouble finding this? Links relevant to quizzes. There's a newly populated link, Papes circuit. And there's another beautiful graphic, which I won't test you on. But it looks more like lollipops, so I'm happier. And here's your hippocampus. And there's a bunch of cells in there that send axons out to form the fornix. We're not going to sweat post-commissural fornix, but I bet you can bet that the post-commissural fornix is behind the commissure. And there's the anterior commissure. And then it goes down into the mammillary bodies where it talks to another cell. And then those axons come up, forming the mammillothalamic tract, meaning it goes from the mammillary bodies to the thalamus. And there it goes to the anterior thalamus, talks to another cell in there, which goes up to the cingulate, which relays one or two more times. And then those axons arc back and around and go back to the hippocampus. Papes circuit. Now, this is occurring on both sides of the brain, obviously left and right. You don't have to draw that, but you do have to draw this. So I crafted this morning. It's very simplified. Here's the cell body inside the hippocampus. It has an axon that arches around and goes down and synapses onto a cell in the mammillary body. And that arcing structure is the fornix. So that's a circle indicating these fibers are the fornix. It's synapses in the mammillary body. Those axons go up to the anterior thalamus, and they go up to the anterior thalamus. That's another circle, lassoing the medial thalamic tract. Oh, no, I'm sorry, the mammillothalamic tract, from the mammillary bodies to the thalamus, mammillothalamic. So mammillary body, to the anterior thalamus, and then they go up to the cingulate gyrus and synapse, and then they arc back around and synapse back on the hippocampus. So that you should be able to draw come next Tuesday. It's an important circuit. And there's one of these on the left side of your brain and one on the right. And consolidation of memory can be very messed up if both of these go away. There will be stories. I will tell them later. So let's look at this one more time. I usually draw it starting in the hippocampus, but of course it's a circuit. It starts in any place. So hippocampus, that's a cell in the hippocampus. Obviously, it has a different shape. Its axon arches around and down and back a little bit to cells in the mammillary bodies. Those cells in the mammillary bodies send their axons pretty much straight up to the anterior thalamus, and that straight up pathway is the the mammillothalamic tract, and then the cells in the anterior thalamus. This doesn't get a name in our story. Go up to the cingulate gyrus, which is just above the corpus callosum, and they synapse on cells there, and possibly there's another link that is shown in the other figures, but I'm not going to use it here. But those axons come back and around and hit the hippocampus. So there you go. Papes circuit. Then, these are important elements of the limbic system. And then we get to the Kluver-Bucy syndrome. These are two researchers, Kluver and Bucy, and I believe primarily working with rhesus monkeys. They started doing lesions of the Papes circuit, and one of their favorite things to do, they weren't specifically doing Papes. They did fairly radical, which means big, radical bilateral, which means both sides, radical bilateral lesions of the temporal lobe. And if you look in your own textbook, if you take out here, you're taking out a lot of stuff. The amygdaloid complex, the part of the hypothalamus, hippocampus, and you get Kluver-Bucy monkeys. No fear, no aggression, strong oral tendencies. They pick something up, it goes right in the mouth, including rocks, yes. Although in the research, I think they did nuts and bolts. And they were charged with being hypersexual. A better description probably is that they were indiscriminate. A horror story here is that physicians went to a conference in Hawaii, saw movies of hypersexual, I think by this time they were doing cats, that were mating with chickens and other cats, and it was just craziness. And then the researchers in the video that the physicians were watching in Hawaii blew up part of the hypothalamus. And these physicians came back from Hawaii and said, oh, we can fix what? Hypersexuality? No, homosexuality. So we will find willing homosexuals who will volunteer for us to fix their male-female balance by blowing up the hypothalamus. It's unbelievable what they got away with. Complete mythology. But fear is important to the circuit, and I mean, you blow up the hypothalamus, you're going to do a million things. Sexual behavior will certainly be affected. Again, every time I see amygdala, I think about amygdaloid complex. And we're about to get a figure from Ledoux, who I'm a big fan of, and I've always wanted to have someone work in his lab. So this is pretty relevant to me. The animals are getting auditory cues, and the cues may be associated with getting shocked. By the way, that grid could not shock them. That's bad graphics. And you get changes in blood pressure and freezing. Now, freezing means the animal doesn't move, which is what rodents tend to do when they think they're about to be munched on. So here they don't move because they've just been hurt. Here they don't move because there's a stimulus associated with just being hurt. And here's Ledoux, and there we go, Ledoux, 1994, 1996. I saw him give this talk at Neuroscience, and I walked out of there, and I have never viewed the amygdala the same way. It's the amygdaloid complex. So if you think back to the earlier parts of this chapter, we had James Lang and Cannon-Bard and Schachter. There's parallels here. So you have stimuli that go to your sensory organs, and then the thalamus, which I must have said 20 times, is the great sensory relay station of the brain through which all sensory information, except olfaction, passes on the way to the cerebral cortex. So sensory information goes into the thalamus. There should be a little lollipop synapse on the cells in the thalamus, and then axons from cells in the thalamus take either the high road or the low road. Now, I found that you guys don't know what this means anymore, but it was a song. You take the high road, and I'll take the low road, and I'll be in Scotland for you. So the low road is faster. And that's why it's called the low road here, because of the song. And by the way, Ledoux has a band, and if you're ever in New York City and you can catch him, which I've never done, I would recommend that. So the low road goes from the thalamus directly to the amygdaloid complex, whereas the high road goes from the thalamus to the sensory cortex and the hippocampus. It gets thought about, and then that evaluation is sent into the amygdaloid complex. And you get different responses. Low roads are faster, obviously. You take the high road, I'll take the low road, I'll get to Scotland before you, for you. By the time the high road figures out what you're looking at, you may have shot someone. Thank you, guns. So in the amygdaloid complex, we have lateral nucleus, which sadly is not so lateral in this figure, central nucleus, which is pretty darn central, and it goes out to emotional behavior, autonomic responses, and hormonal responses. And in this edition of your book, they have dropped my beloved periaqueductal gray, which is in one of those eras, but I won't burden you with that. Let's look up Ledoux really fast. And his band is the Amygdaloids. And there's the band. So there he is. These are graduate students, and they play in bars in New York City. And let's play this one. And this looks like our part of the world, doesn't it? I mean, he's in New York City. Maybe the Poconos. Nice red boots.
While I was fearing it came, but with less of the fear, because fearing it so long had almost made it dear.
Oh, he's not intense and scary, but he's brilliant. And he writes books for the popular press and does cutting edge research. Big fan. So it's Ledoux, it's the amygdaloids. Enjoy if you wish. So he teaches you about neuroscience through music. By the way, this figure over here gives you a better 3D representation of the hippocampus and fornix and mammillary bodies. You know, hippocampus is pretty lateral. Mammillary bodies are right in the middle. So remember the banana? It's going to arch in, and then the fornix, and then the mammillary bodies. That's a good figure. Spend some quality time looking at that. Uh, see here, healthy. See here, oh my God, where did the amygdala go? It's destroyed. And there's different stimuli and how controls reacted and how SM reacted. Uh, Very different. No surprise, emotions affect different parts of the brain. Um, and don't obsess over this, but do learn the parts: orbital frontal, prefrontal cortex. You want that functioning pretty well if you want to have good adult decision making processes. Singular cortex is all this. Looks like a C, looks like a banana. It's over the corpus callosum. Anterior, which means it's in front of the posterior. That's pretty good. Here's another view of the orbital frontal, right over your eye sockets. We haven't talked much at all about the insula, and I feel kind of bad about that, but it's important here. There's the anterior cingulate again. There's the amygdaloid complex again. Um, so you arguably you should be able to identify what you're looking at in any of those pictures. And there's major emotions and scans and colors. And I'm not going to test you on that, but do take a look at it. What is aggression? There are different types of aggression. Um, and I did research on this as an undergraduate. I forgot. Uh, Males are bad. Uh, they're very aggressive. They fight with each other. Uh, my mice sometimes fought with each other unto death, even though we didn't ask them to. Androgens seem to increase aggression within limits. Testosterone, uh, commonly in folks. So you want to make males less aggressive, castrate them. And there are data. And then if you say, oops, that was a mistake, I shouldn't have castrated you, I can give you back your testosterone and you'll get pretty normal again. And sexual behavior will go with that too. Dominance behavior. Women treated with testosterone spend more time staring into the eyes of threatening faces. You looking at me? You looking at me? I made eye contact with a guy in the bar, and he almost killed me. Uh, you do not make eye contact with alpha males unless you want to fight. And I didn't. I just was being friendly. Um, this on the right, and GABA is discussed in here, and serotonin. A lot of things are important, obviously. And there's your ventral medial hypothalamus, which I don't know. It's come up in 3 or 4 chapters now. Biopsychology of human violence, a topic of controversy. Emotional discontrol syndrome, psychopaths who freak me out. And that picture of Ted Bundy, that was in the courtroom. Uh, wow, wow. Uh, that induces fear in me. Um, I'm not a fan of psychopaths, although I'm studying them. Um, and stress. So I've done a fair amount of stress research. There's a pathway: faster response, slower response, high road, low road, sort of, but not the same. Take a look at that. And there's jumping off a plane. And there's a review of your sympathetic and parasympathetic nervous system. There's the sympathetic chain paralleling the cerebral cortex. I'm sorry, the spinal cord. And you have parasympathetic outputs from your brainstem and your sacral cord. And you had this way back in chapter nothing. It's back comprehensive final. And then there's cortisol, testosterone, epinephrine, norepinephrine, growth hormone. All sorts of things are affected by stress. And social stress, I can relate that. Gasoline rationing, I remember that. We did that one. Um, and stress immunization. This again reminds me of Dr. Karpiak and doing research to make adult mice happier based on earlier childhood type experiences. And a little stress early in life seems to make animals more resilient to later stresses. So, you don't want your children to grow up to be snowflakes that can't handle any stress. If you're living at home with mommy and daddy, that'll work out fine, I guess. But boy, you go out in the real world like that and you get slaughtered. Genetics, which has come up several times in the book, is in this chapter. And there's stress and emotions affect the immune system. This was just beginning to be researched as I left UCLA. Psychoneuroimmunology, which is one of the reasons our lab is involved in promoting pain as the fifth vital sign, because pain is a stressor. Stressors can suppress the immune system. Suppressed immune systems in injured people means greater likelihood of secondary infections. So please do not get rid of the concern over pain as you deal with the opiate crisis. And this is cute. People who felt they had little or no sense of purpose in their lives, time since they were diagnosed. No sense of purpose, you die. There's psychoimmunology in the 80s. That's 80s is when I came here. They were just starting to do that as I left UCLA. And there's your phagocytes, hopefully attacking something appropriate. And you have to learn these terms. You just have to learn these terms. And my immune system is not doing me any favors right now. So take a look at this. Interaction between neurosystems and immunosystems. It was controversial in the 80s when this started, but boy, we believe it now. And good graphics, more than good graphics, explaining how things work, and stress and immunity. You may notice that you get more infections around exam times because of stress. And there's a discussion of that. There we go. Stressful exam periods, decline in number of immune cells. Why does stress suppress the immune system? We'd like it not, but it can. And mindfulness, which is related to cognitive behavioral therapy, the therapy that works. Mindfulness may be able to help reduce immune suppression. I'm not going to test you on this table, but it's a nice overview of things that acute stress, we're made for that. Prolonged stress, not so much. And oh boy, that's brilliant research. That's a live mouse anesthetized, but you're visualizing those dendrites right through a portal in the skull, and you can see things changing. Let me. This is the end of the chapter. So, I think. Hold on. Yes. See this guy here, Sapolsky. Why zebras don't get ulcers. Sapolsky. Zebras don't get ulcers. I love this guy. I like a lot of researchers. So you can buy this book on Amazon if you want for not too much money. A clean gut distress, stress-related diseases and coping. He's at Berkeley or Stanford. I always get confused on those two with him. He's got some brilliant online lectures about neuroscience in general and stress in particular. How much do I love him? Let's go to Tim Cannon's page, Tim Cannon's homepage, and let's go to the pull down and Behave Clavius. The school has established a fund to fund faculty getting together from different disciplines to talk about important books. And 2 summers ago, this was my book. And I ran a Clavius, and there he is. In neuroscience, he usually wears very bright floral shirts. And here are Sapolsky links. And the book that we were talking about is this one. It's called Behave. Behave, the biology of humans at our best and our worst. That was our discussion book. And what a brilliant title, Insight into the Brain, in a book you wish you had in college. Well, I'm not selling it, but I'm opening it up to you. This is a great book. And it's, at times it's heavy neuroscience, but he writes really well. He views the world in a way that there is right there, Behave. So a whole bunch of faculty last year spent the entire year talking about that book. Oh yeah, okay. So there's a TED Talk by him. The MacArthur Fellow at 30. He was named the MacArthur Genius. There, he has a wiki page, and obviously he's at Stanford, not Berkeley. Carleton College must have more money than God. They paid him to come and give a talk, and there's a talk. And on Facebook, if you're familiar with it, there's a Robert Sapolsky fan club, which I don't spend a lot of time on. It's unofficial. Can't read it. I love science, and it's something. Can't read it. I love science, and it pains me to think that so many are terrified of the subject, or feel that choosing science means you cannot also choose compassion or the arts, or to be awed by nature. Science is not meant to cure us of mystery, but to reinvent it and reinvigorate it. Amen and amen. So he's an impressive dude. I like Sapolsky. Okay, so I have blasted through this chapter, so if they lock me up in the hospital, you have a lecture. I just have to figure out where the turn off thing is. Right here. Okay, no, where's the turn off? How about over here? So good day. I hope I'm doing this